Okay, we want to have a discussion here about the lymphatic system, and we're going to actually couple that with a discussion of the lymphatic system coming up in our, our next podcast. Okay, the lymphatic system's a second circulatory system in the body whose purpose is to return interstitial fluid lost to capillary beds to a system of vessels that will take that fluid and filter it through structures that are called lymph nodes and then return that filtered fluid to the blood at the right and left subclavian veins. Other lymphatic organs include the thymus, the spleen, the tonsils, mucosa associated lymphatic tissue. Okay, and these all have roles in promoting immunity and removing pathogens from the blood and from the body fluids. Okay? About 15% of the fluid volume is lost at every capillary bed in the body and gets filtered in the lymphatic system and returned to the bloodstream. And if this didn't happen, the buildup of fluid in those tissues would cause a condition known as edema. There's three parts. There are lymphatic vessels whose flow is one way back towards ultimately the heart via the circulatory system. There's the lymph, which is the fluid in the vessels. It's the other liquid connective tissue along with blood in the body. And lymph nodes, whose job it is to filter and clean the lymph by removing cancer cells and other types of pathogens, both living and non-living. The lymphoid organs are the structural basis for the immune system, and they house macrophages and lymphocytes, which, as we learn in the immune chapter, have to work together in order to promote defense against disease based on the presence of non-self antigens, which are molecules on the surface of a pathogen that are unlike our own. And so that tags them chemically for destruction. And these can be really anything on the membrane surface or on the surface of the pathogen. They could be a glycoprotein, they could be a lipid, a lipoprotein, really almost any combination of molecules on the surface has the potential to be a non-self antigen. The other structures are the spleen, thymus, tonsils, and lymphoid tissues. The lymphatic vessels return interstitial fluid and leak plasma protein back to the blood at a rate of about 3 liters daily. And once the interstitial fluid goes into the lymphatics, we call it lymph. And you can see here that it's at the capillary ends of the circulatory system that this is going to occur, right? We have blood flowing in through arterioles into capillary beds, and we lose quite a bit of it via both hydrostatic pressure and osmotic pressure to the surrounding tissue at the arterial end, and we recover quite a bit of it using the same forces at the venual end, but we don't recover all of it. And that 15% that resides and is left behind in the tissue fluid is what's going to produce interstitial fluid pressure that will open one-way valves in the lymphatic vessels and allow passage of this fluid into the lymphatic system. It is a one-way system that always promotes flow towards the heart. The lymphatics include the lymphatic capillaries and the collecting vessels, and the lymphatic trunks and ducts, and so that's from small to large, right? The lymphatic capillaries are a lot like blood capillaries, except that they're more permeable. As a result, they can take up large proteins and even whole cells and cell debris, and that's what we want. The endothelial cells overlap and form one-way valves that are anchored by collagen filaments, preventing the collapse of the capillaries. And it's the increase in extracellular fluid volume, the interstitial fluid, that is going to open those mini valves up. And then once the pressure inside the vessel is greater than the extracellular fluid pressure, the valves will shut. The pathogens travel through the body using the lymphatics, and we don't find lymphatics in bones, teeth, bone marrow, and in the central nervous system, although there is some evidence that there may be some lymphatic drainage from bone, okay, and that's relatively recent. The lacteals are lymph capillaries that are in the intestinal mucosa embedded in structures known as villi, and their job is to absorb dietary fat and deliver that chyle to the blood via drainage into the cisterna chyli and then into the thoracic duct, and then from there into the left subclavian vein. Okay? So you can see here how it works. When the pressure builds sufficiently outside the lymphatic vessel, we force the flaps open. The fluid goes in one direction, down its pressure gradient, then it gets trapped inside the vessel, and then as the pressure builds up to be equal or greater than the extracellular fluid pressure, the one-way valves will shut, and then we will start the transport of lymph through these vessels using a combination of one-way valves, skeletal muscle, and respiratory pumps, and a little bit of smooth muscle that's also in the lymphatics. Okay? The collecting vessels are a lot like veins, except that with thinner walls and more valves, and they anastomose more frequently. Anastomosis, remember, is the fusing of two or more structures into fewer structures. It's the opposite of branching. They are going to end up as collecting vessels in the skin, traveling with superficial veins, usually also tracing the course of a nerve in many cases. The deep vessels are going to travel with the arteries and also probably with a nearby nerve. Nutrients are supplied from the vasa vasorum, and remember that those are blood vessels that actually supply nutrients to the walls of other vessels. Okay, so vessels that feed vessels. The lymphatic trunks are formed by the union of the largest collecting ducts, which are the paired lumbar, the paired bronchiomediastinal, and the paired subclavian and jugular trunks. Okay? There's a single intestinal trunk as well. Lymph is delivered into one of two large ducts. The right lymphatic duct drains the right upper arm and the right side of the head and the thorax, while the thoracic duct comes from the cisterna chyli, which is around the lumbar spine on the anterior aspect, and drains the rest of the body. So our lymphatic drainage is not symmetrical. Okay? Each empties lymph into a venous circulatory subclavian vein, okay, on its own side of the body. And so you can see the difference, right? The green is what the right lymphatic duct will drain, okay, and the rest of the body is what the thoracic will drain, okay? So non-symmetrical. Why that is, I really couldn't tell you. It just is. And you can see here the flow, right? Here is the cisterna chyli collecting nutrient-rich fluid via the lacteals in the villi of the digestive system. The chyle, which is the fatty lymph, collects here and then moves up the thoracic duct and eventually is going to enter the left subclavian vein, right? And then on the right side of the body, the right pectoral region, the right shoulder, neck, head, and arm are going to enter via the right lymphatic duct, right in there. Okay, so the fluid gets recovered. Okay, unless there's something that's blocking the circulation. Lymph is moved through the body by the milking action of skeletal muscle and pressure changes in the thorax that take place when we breathe, as well as one-way valves preventing backflow. Okay, and pulsations of nearby arteries help out, as well as the smooth muscle in the walls of the lymphatics. Okay, so let's look at the dynamics of flow here. Okay, the lymphoid cells, which are basically agents of our adaptive immunity, include the lymphocytes and the macrophages. Okay, all of these originate in the red bone marrow, which is primarily found in the axial skeleton. But they can be divided into two varieties, which are the T cells, who finish their maturation in the thymus, which is behind the sternum and in front of the heart, and the B cells, which finish their maturation in the red bone marrow. The B cells are selected and produced daily in the red bone marrow, while the T cells are selected and distributed in basically one big event where they move out of the thymus and they take up residence in places like the tonsils and the malt and the spleen and the lymph nodes, okay, and in the dermis of the skin, all right, and they remain there and wait to encounter the antigen that they are designed to bind to, okay, so they're sort of there doing a stakeout, right? Looking for the bad guys. T cells and B cells are designed to respond to foreign antigens. These include bacterial toxins, bacterial cell wall molecules, which could be proteins, could be polysaccharides, could be lipids, okay? Structures on the surfaces of viruses, red blood cells that bear the wrong antigens, and cancer cells whose antigens often change as they transition from a normal to a cancer cell. So the antigen itself is usually not what presents a danger to the body. It's actually the pathogen to which it's attached. And remember that pathogens can be living or non-living. A living pathogen would be something like a bacteria or a parasite, or in some cases a mold or a fungus. Okay, and then there's even examples of pathogens such as nematodes, right? Little little worms that can get into the body through a variety of ways. Okay. Um, Non-living pathogens would include things like toxins, prions, and viruses. And I can say that they're non-living because they don't have the ability on their own to reproduce. They have to borrow machinery from the body in order to do that. A T cell is designed to attack and destroy cells that are infected, which indicate that they're infected by presenting non-self antigen on its surface along with a special membrane protein called an MHC1. Okay? While a B cell is going to produce antibodies by transitioning into a plasma cell after it has first encountered the antigen to which it binds and then gets fully activated by interaction with a helper T cell. And then it will begin to produce antibodies, which are proteins that drift through the plasma and the interstitial fluid and lymph and bind to the antigen wherever it is found and tag it for destruction in a variety of ways that are detailed in the chapter on immunity. Okay? Macrophages phagocytize foreign substances, and they help to activate T cells by presenting the foreign antigen to them on an MHC2 protein. And it's the T cell, the helper T cell specifically, that helps to fully activate the B cells and the cytotoxic T cells. Dendritic cells capture antigens and deliver them to lymph nodes, and reticular cells make reticular fibers that support the other cells in the lymphoid organs. So they really don't have an immune function, but they kind of build the superstructure. And you can see what these look like here, right? You can see the macrophages and the reticular fibers of lymphocytes and all these fluid-filled spaces through which the antigens are going to flow, right? Lymphoid tissue provides a site for the reproduction of lymphocytes, as well as macrophages, as well as a site for the surveillance of pathogens that flow past them. Reticular connective tissue is loose connective tissue that has fluid-filled spaces in it, and it's those fluid-filled spaces where the pathogens may interact with the macrophages and the lymphocytes, be identified, and then that will promote the reproduction of lymphocytes that are specifically designed to bind, attack, and destroy either infected cells or pathogen that is yet to enter a cell in a tissue. Okay? So if we were to sort of have a division of labor here, the cytotoxic T cells are the search and destroy for infected cells, right? The infected cell says it's infected by showing a foreign antigen, so we kill it. And the B cells make antibodies that are designed to catch the pathogen before it gets in and tag it for destruction. Okay? So B cell, the pathogen's out in the open, and T cell, the pathogen is inside one of our cells. Okay? Okay, now with lymphoid tissue, we have different types, right? We have capsulated and unencapsulated lymphoid tissue. An example of capsulated lymphoid tissue would be lymph nodes. Okay, unencapsulated would be something like malt. All right. Diffuse lymphoid tissue and lymphoid cells and reticular fibers are in almost every body organ. There's a lot in the lamina propria of mucous membranes for a very simple reason. Number one, lamina propria is loose connective tissue, which allows for the movement of these white blood cells so that they can more effectively fight pathogens. But number two, okay, the lamina propria is generally going to be in the walls of body systems that are exposed to pathogens from the outside environment, such as in the digestive system. Lymphoid follicles are solid spherical bodies of tightly packed lymphoid cells and reticular fibers. The germinal centers are for proliferating B cells, so rapidly dividing B cells, and can form part of a larger lymphoid organ. And then there are aggregations of Peyer's patches in the ileum and in the appendix because of the large concentration of bacteria that reside in the colon. The lymphoid organs of the body are the lymph nodes, the primary lymphoid organs. And they're embedded in connective tissue in clusters along lymphatic vessels concentrated in the inguinal, axillary, and the cervical regions. And this is for a very important purpose, right? Those areas are near places that are exposed to the outside, right? If you think about, for instance, the inguinal region, there's at least 2 openings down there in both men and women, and in women there are 3 openings in the inguinal region, right? Which means that pathogens can get in from the outside. That's a port of entry. So it makes sense to cluster the lymph nodes where they will drain from organ systems that are exposed. Okay, axillary region. What do we have in the axillary region? Well, we have the respiratory system, okay, nearby, and we also have the openings of the mammary glands at the nipples. Okay, and again, those are ports of entry. Not to mention the esophagus. Okay, as another example. Cervical region. All right, up in the neck. Why so many? Well, think. You've got your eyes, your nose, your ears, your mouth, all ports of entry. Okay, so it makes sense to put the filtering units where they're going to be the most effective. So they filter the lymph with the macrophages destroying the microbes and the debris, but the macrophages have a second role, which is to present the antigens from that destruction to the lymphocytes, which then bind to those antigens and get activated and multiply and then go out and destroy the antigen wherever they find it attached to something. Okay? So macrophages and lymphocytes always have to work together in order to be effective. Okay? Macrophages are a class of cell that are called an antigen presenting cell, meaning their job is to chew up the invader and kill him, and then take pieces of it and show it off to the lymphocyte population in order to activate them. Okay? So there's your drainage, right? You can notice where the bulk of the lymph nodes are clustered in the regions that we indicated, okay? And if you get blockage in any of these regions, you're going to get edema upstream of the drainage, okay? So for instance, if I block the inguinal lymph nodes, you get massive swelling in the scrotum. If I block the popliteal lymph nodes, you get massive swelling in the lower leg, and so on. Lymph nodes vary in shape, but they're about bean-shaped with a capsule outside and trabeculae that extend inward, dividing it into different lobes, right? The regions include the cortex, the medulla, okay? Cortex being the outer portion, and the medulla being more towards the core. And the idea, again, is that fluid percolates through this, and as it does, it interacts with both the macrophages and the lymphocytes. The cortex contains follicles with germinal centers, and that's where a lot of the B cells get produced. The dendritic cells are going to encapsulate the follicles, and the deep cortex houses the T cells that are in transit, circulating continuously between the blood, the lymph nodes, and the lymph. So they're basically marching around, they're doing their job. The medullary cords extend inward from the cortex and have B cells, T cells, and plasma cells, okay, mature ones. The lymph sinuses are primarily populated with macrophages. Okay? Remember that a sinus is just an open area inside an organ. It can contain either gas or fluid or both. Lymph is going to enter the convex side of the lymph node through afferent lymphatic vessels. Again, this is a one-way movement through the filtering unit. Then travel through subcapsular sinuses and smaller sinuses to the medullary sinuses, and then exits the concave side at the hilum through the efferent vessels. Okay, so there are more afferent than efferent vessels, and that's to slow the flow sufficiently so that we have time for the lymphocytes and the macrophages to do their work. Okay, if the material goes through too quickly, then we're going to have a problem effectively catching, destroying, and presenting the antigens from the pathogens to the lymphocytes so that they can be activated. And so that's exactly what we note here, right? So you can see here the different parts of the lymph node, right? Those here, let's do the laser again. So laser pointer. These are the efferents, right? And these are the afferents. Okay, pretty straightforward. And then there's a photomicrograph. Okay. But again, you can see, right, there's the capsule and the trabeculae, right? That's the non-immune part, okay? And then you got cortex out here, and there's your germinal centers, okay? This is medulla, and all this out here, this is cortex, okay? The spleen's job is to remove pathogens from the blood itself and to aid in the recycling of worn out red blood cells. So another one of its roles is to act in the recycling of things like amino acids from the hemoglobin and iron from the heme. Okay? It's served by the splenic artery and vein. Remember that the spleen is on the left side of your abdominal pelvic cavity under your diaphragm at the end of the tail of the pancreas. This is also an area where lymphocyte proliferation and immune surveillance can occur. But again, what's going on in this immune surveillance is we're actually checking out the blood as opposed to the lymph. So it cleans the blood of aged cells and platelets, and the macrophages remove debris, and of course we pick off pathogens as well. And there's your spleen. Okay. It superficially, if you just glance at it real quick, looks a little bit like a kidney, but that's only because of the robust blood supply and the general shape. Okay. The spleen stores breakdown products of red blood cells, such as iron for later use. It stores platelets and monocytes, and it is the site of fetal erythrocyte production until the red bone marrow takes that over at around birth. Okay. It's enclosed by a capsule, and it has trabeculae just like the lymph nodes do. And it, of course, has lymphocytes and macrophages and lots of red blood cells. And you can see a cross section of the spleen here, right? The two distinct areas are the white pulp around the central arteries, which are mostly lymphocytes on reticular fibers, and they aid in immune function. And then the red pulp in the venous sinuses and the splenic cords is high in red blood cells and macrophages for disposal of worn out erythrocytes, bloodborne pathogens, and other toxins. And it's composed of splenic cords and sinusoids. Okay? The thymus is critical early in life because it's the site of maturation of our T lymphocytes. It's found in the inferior neck behind the sternum and in front of the heart. It increases in size and is most active during childhood, and then it stops growing during adolescence and gradually will atrophy. It'll still produce immunocompetent cells, but it'll do so rather slowly. Thymic lobules contain outer cortex and inner medulla, and most thymic cells are lymphocytes. The cortex is going to have rapidly dividing lymphocytes and scattered macrophages as well. The medulla contains fewer lymphocytes and thymic corpuscles involved in regulatory T cell development in order to prevent autoimmunity, which is where the immune system attacks our own tissue and destroys it. And it differs from other lymphoid organs in that it has no follicles because there are no B cells here, and it doesn't directly fight antigens. Instead, the job of the thymus is to determine which T lymphocytes are going to serve the job of recognizing and destroying anything attached to foreign antigen while tolerating our own antigens. Okay, so those cells that don't meet that set of criteria are either destroyed or they're rendered in a state of anergy, in which they're alive but they don't participate in the immune system. And so when you're doing that process, you don't want anything interfering with it, and so that's why we have this blood-thymus barrier that ensures that while we're essentially schooling the T cell population, we don't have interference from any outside molecules. The stroma of epithelial cells provides the environment where the T cells are rendered immunocompetent. And here you can see the capsule and cortex and medulla and the thymic corpuscles, right? And so what will happen is that once the T cells have been selected, right, we will have that population graduate from the thymus and move out and populate the lymph nodes and the tonsils and the malt and all the other places that we expect to find T cells. At that point, the thymus is less important and it begins to atrophy. Lymphoid tissue is in most mucous membranes in the body because that's going to be exposed to where pathogens can come in. So it keeps the pathogens from getting into the blood. That's the big objective. The largest collection of malt is found in the tonsils and the Peyer's patches and appendix. It's also in the mucosa of the respiratory and urogenital system and in the digestive tract. The tonsils are simple lymphoid organs, and they form a ring of lymphatic tissue around your throat. The palatine tonsils are at the back end of the oral cavity. The linguals are grouped at the base of the tongue, and the pharyngeal tonsil is in the back wall of the nasopharynx. Tubal tonsils surround the openings of the auditory tubes into the pharynx, and they gather and remove pathogens in food and air, and they have follicles with germinal centers in them and aren't fully encapsulated. Overlying epithelia is going to have these little indentations in them that are called tonsillar crypts, and the idea is that you get pathogens in there that are trapped in these deep crypts, and then the macrophages and lymphocytes move in and destroy it. And this allows us to build memory for other pathogens, and it also intercepts a lot of the pathogens that try to make it in through ingestion or inhalation. And again, the tonsils don't always win the battle, right? The tonsils can, in some cases, lose the battle, become inflamed, and that's called tonsillitis. And when that occurs, we generally surgically remove the tonsils because at that point they become a source of infection rather than a defense against it. The Peyer's patches, which are shown here, are clusters of lymphoid follicles in the wall of the distal small intestine, primarily the ileum. Similar structures are also found in the appendix. They destroy bacteria and they prevent them from breaking through the intestinal wall, and they generate memory in lymphocytes so that the next time we encounter that antigen, it's easier to destroy because we already have a waiting army that can attack anything that antigen's attached to much more effectively. Now, where does the development of the immune system start and stop? Well, the answer to where it stops is it never really does stop. Right? It continues throughout life because you're making B cells every day of your life. Okay? But it begins with clusters of lymph nodes in the 5th week of embryonic development that come from lymph sacs in developing veins. The jugular lymph sacs then form the right lymphatic duct and thoracic duct, and then lymphatic organs, with the exception of the thymus, come primarily from mesoderm. Lymphoid organs, with the exception of the thymus, develop from mesodermal mesenchymal cells. The thymus is endoderm and is an outgrowth of your throat. Except for the spleen and tonsils, the lymphoid organs are not well developed at birth, which is why we promote nursing from mother's milk as a way to acquire passive immunity, as well as the passive immunity acquired from the mother's antibodies that cross the placenta and get into the baby's bloodstream. And so what they do is they sort of form a shield against infection while the baby's own immune system is becoming more effective. After birth, high numbers of lymphocytes in their development parallels the maturation of the immune system. And again, it's not until the T cells get deployed from the thymus that we're really in what I would consider to be the adult state of the immune system, right? Where we've generated an immunocompetent population of T lymphocytes. And it's going to be in the immune chapter where we're going to talk more in detail about what the different types of white blood cells do. Okay, we've talked a little bit about it in the blood chapter, but we're going to get more in depth with the lymphocytes and the macrophages and the granulocytes in order to understand why it is that when we live in a world that's filled with pathogens, we spend most of our time relatively healthy, which is sort of amazing. Now, what follows this is a little discussion of the immune system, sort of a summary, okay? And then I will join everybody in the next podcast. And I thank you for listening.
The lymphatic system. Lymph vessels are found in all tissues except the central nervous system, the bone marrow, and tissues without blood vessels such as cartilage. The lymph system vessels are extensive as the vessels of the circulatory system. The lymphatic system serves several functions. It controls fluid balance by draining and cleansing the fluids that leave the circulatory system to deliver nutrients and gases to the tissues. It interacts with the villi in the digestive system to absorb and deliver fats to the circulatory system. It also has an immunological protection from viruses, bacteria, fungi, and cellular debris that could damage the cells of the body. From your understanding of the circulatory system, you know that the blood passes through the arteries, arterioles, and then the capillaries. The capillary walls allow the fluid portion of the blood to exit the capillaries into the surrounding tissues. Once the fluid leaves the capillaries, it is called interstitial fluid. About 90% of this fluid will diffuse back into the capillaries because of the difference in concentrations of the fluid. However, about 10% of the fluid will enter the open-ended lymph vessels. Once the fluid has entered the lymph vessels, it's now called lymph. These vessels eventually deliver the lymph to locations where the lymph can be cleansed of debris and checked for the presence of pathogenic organisms. How it gets the lymph there is pretty amazing. There is no heart for this system of vessels to pump the lymph around. So how does the lymph get to the locations it needs to be delivered to? The lymph moves through your body when you move your skeletal muscles. The contraction of skeletal muscles squeezes the nearby lymph vessels, pumping them. This pushes lymph through the vessels. In addition to the contraction of skeletal muscles, there are two other means by which lymph travels through the lymphatic system. There are smooth muscles at the larger lymph vessels. The contraction of these smooth muscles adds to the force provided by the skeletal muscles. Also, when we breathe, pressure changes occur in the thoracic region. When the thoracic pressure drops, that tends to pull lymph into the thoracic duct. One-way valves prevent the lymph from flowing backwards. The function of fluid balance is seen best, perhaps, when it goes awry. When the lymphatic system is prevented from doing its job, the fluids build up in the tissues. Edemas is the term given to this medical condition. Mild edema can occur during pregnancy when the weight of the baby slows the ability of the vessels to move the lymph up the body. More serious levels of edema can occur in a tropical disease called elephantitis, in which a parasite blocks the vessels, and the edema that is produced looks a lot like having legs of an elephant. Some lymph tissue is very diffuse, with no clear boundaries. You can actually feel some when you rub your lower inner lip with your tongue. Others are more organized into groups, and these are called lymph nodes. Lymph nodes have three functions. First, they are testing stations. They monitor the blood by receiving samples of the blood plasma. Second, if the sample is rife with foreign invaders, they produce lymphocytes and send them into the bloodstream to try to destroy those invaders. In addition, the lymph nodes filter the lymph that they have so they can only return clean fluid back to the blood. Eventually, the lymph is returned to the circulatory system via the right and the left subclavian veins in the shoulders just above the heart level. Lymph nodules can be found as single structures in the body, or they can be grouped together in small clumps. That's what the tonsils are. They're groups of lymph nodules under the mucous membrane in the throat. These lymph nodules form a protective ring around the throat strategically located to protect the body from foreign invaders. If the tonsils get infected, they can become inflamed and abnormally enlarged, as you see here. This condition is called tonsillitis. If the condition is chronic, the tonsils can be removed in a tonsillectomy. Tonsils tend to get smaller as a person matures, and they can actually disappear altogether in an adult. Peyer's patches are very similar to tonsils. They are groups of lymphocytes and lymph nodules that are in the small intestines. Typically, they're found in the last third of the small intestine. Once again, they're strategically located to deal with foreign invaders. The lymphatic system's second function takes place here in the small intestine as well, the absorption of fats. We will discuss this more in depth in the topic of digestion, but for now, know that there are specialized lymph vessels called lacteals in the intestinal villi. These pick up fats that are released from digested food and absorb it into the villus tissue. The liquid in the vessels take on a milky color. Instead of being called lymph, this fluid is called chyle. The chyle eventually gets dumped in the subclavian vein, just like lymph. That is how the fats enter the circulatory system. The spleen is a significant lymphatic structure and has a lot in common with the smaller nodes throughout the body. But unlike the lymph nodes, the spleen does not filter lymph. It's part of the lymphatic system, however, because it filters the blood. As the blood passes through the white pulp of the spleen, foreign invaders stimulate a response from the diffuse lymphatic tissue or the lymph nodules. The spleen also works to clean the blood of worn-out erythrocytes. Remember, red blood cells have a short lifespan. As a result, roughly 2 million erythrocytes die every second. They must be removed from the blood, and that's another job of the spleen. Before the blood leaves the spleen through the veins, it passes through the red pulp. Macrophages in the red pulp engage in phagocytosis to remove both foreign invaders and worn-out red blood cells. The third function of the spleen is to act as a reservoir for oxygen-rich blood. The spleen actually holds more blood than is necessary for its own metabolism. Therefore, it's an extra blood supply full of oxygen and nutrients. This serves as a backup supply of blood in case of blood loss. If the body detects blood loss due to hemorrhage, the sympathetic division of the ANS stimulates the smooth muscles in the capsule of the spleen to contract. This pushes the backup supply of blood into the bloodstream, compensating for the blood loss. Although the backup supply of blood in the human spleen is rather minor, it's a major factor in the physiology of some other mammals. Seals use the spleen as a built-in oxygen tank. When the seal dives, it conserves its oxygen as much as possible. However, when it's running low and cannot get to the surface, the smooth muscles of the spleen contract, sending the oxygen-rich blood stored there into the bloodstream. This gives the seal more time before it must surface to breathe. Although the spleen is part of the lymphatic system, you can live without it. If your spleen is ruptured due to an injury, it can be removed in a splenectomy. This is often necessary in order to stop internal bleeding because the spleen is so vascular. Once your spleen is removed, tissues in the liver as well as other lymphatic tissues in the body take over the first two tasks of the spleen. Of course, the overall function is not as good as when the spleen was present in the body. As a result, people who have their spleens removed are more susceptible to infections and more sensitive to hemorrhage. The spleen is roughly the size of a clenched fist. Unlike lymph nodes, however, the capsule or outer cover of the spleen contains smooth muscle tissue. Extensions of this capsule, called trabeculae, make up the skeleton of the node. The lymph nodes are fed by several afferent lymph vessels. However, lymph exits through just one efferent lymph vessel. Reticular fibers extend from the trabeculae, forming a net of connective tissue throughout the lymph node. Inside the spleen, there are two types of tissue, red pulp and white pulp. The white pulp is composed of diffuse lymphatic tissue and lymph nodules, much like the lymph node. This white pulp surrounds the arteries which enter the spleen. The red pulp is made of twisted veins and reticular fibers which are full of blood cells which were in the capillaries of the spleen. Lymph nodules contain germinal centers where rapid mitosis of lymphocytes can take place in response to foreign invaders found in the lymph. Lymphocytes produced in the germinal centers are released into the lymph and eventually reach the bloodstream, where they can be transported to the tissues. Another lymphatic system structure is the thymus gland. Like the tonsils, the thymus gland changes as a person matures. When a person is young, the thymus gland is large in proportion to the body size. During this stage of life, it is mostly lymphatic tissue. After puberty, it decreases in size and becomes mostly fibrous and fatty tissue. What does the thymus gland do? Like many things in the human body, the scientific community is still rather puzzled by the thymus gland. We know that while a person is young, immature lymphocytes known as T lymphocytes leave the bone marrow. Remember, blood cells are made in the bone marrow, and they travel to the thymus. Through a remarkable maturation process, sometimes referred to as thymic education, T lymphocytes that are beneficial to the immune system are spared, while T lymphocytes that might evoke a detrimental immunological response are eliminated. For example, if you have type A blood, T lymphocytes which attack the A antigen are destroyed. However, T lymphocytes which attack the B antigen are allowed to mature and enter the bloodstream. Notice that this one is called a gland. That means that one of its functions is to secrete hormones, making it also a part of the endocrine system as well as the lymphatic system. It produces hormones. Principal among them is the hormone thymosin. What does thymosin in the body do? Well, we're not really sure. We know that it affects the immunological response of the body. However, the way that it's done remains unclear. One prevalent thought is that thymosin stimulates the activity of lymphocytes to migrate to other lymphatic tissues.